There's no time like the present to finish material from the rest of this chapter. If you watch this and take notes on it, this is the end of the material that will be on the test, and you should, if you're comfortable with all of this material, be pretty prepared for a test on Tuesday. Feel free to take notes and, of course, come in and ask me any questions that you have about any of the material that you're not sure about or you don't quite understand. So after chemical evolution and the processes that would have led to biomolecules forming, the next step would have been biological evolution. So if the heterotroph hypothesis, or Ober and Haldane hypothesis, or primordial soup hypothesis, whichever one you want to call it, if that was correct, then this is what would have happened next. The very first cells. The idea of the very first cells, there's a few scientists who have developed different hypotheses. One of them is Carl Woese. And Carl Woese, E-S-E, Carl Woese said the very first of these early cells would have been called protocells, and that's pretty well accepted now that we call the first things that we think are close enough to life on Earth, we call it a protocell. That they would have been much more similar to something like a virus than a modern-day cell. And the reason that he said it would have been a virus is because it would have been very, very simple. It would have had that self-replicating RNA material as its genetic information, so all of its genetic material would have been RNA, and it would have had an outer coat. That outer coat could have been either lipid or protein. Most scientists now think it would have been a double-layer lipid. And you can see in this particular diagram, they're pointing to a vesicle, which has a double layer right there, in between those two arrows, has a double layer lipid membrane. And this membrane is what establishes the inside of the protocell versus the outside of the protocell, which is kind of important for establishing boundaries of what that living thing would have been, so how large it would have been. It would have been very simple, and scientists accept this as one of the potential early life forms, or those very first quote-unquote cells or protocells, because these lipid membranes do have the ability to divide to make two smaller vesicles. And if that RNA had self-catalyzed its own synthesis, so it would have self-catalyzed its own reproduction, and the vesicle would have split, that is reproduction. So that is making more similar or identical organisms to oneself, and that would have been life. These first protocells would resemble what we consider modern-day prokaryotes. Remember from our classification chapter, a prokaryotic cell, they're the very simple ones. All they really contain is usually modern-day prokaryotes contain circular DNA. They don't have any organelles inside of them other than very simple, very small ribosomes. So there are no mitochondria, there are no chloroplasts. They're very, very simple, and they're very, very small. So protocells would have resembled modern-day prokaryotes. The fossil evidence that we have supporting Carl Woese's protocells, beyond, of course, the modern-day actual observation of protocells in labs, the fossil evidence we have is 3,500 million years old. That's the very, very first evidence we have of early life forms. It's the same age as the oldest rocks that we know, so it's the same era. Any evidence of protocells that would have developed before that would have been destroyed in the early Earth. There's no way that evidence could have possibly survived. But those 3,500 million year old microfossils, we call them microfossils for fairly obvious reasons, micro meaning small, and if they're individual protocells, they are exceptionally tiny. Remember we said they resembled prokaryotes, and when we talked about prokaryotes in classification, we said that they're, very, very small, 5 to 10 micrometers, not anything much larger than that. So these microfossils are similar to prokaryotes and would have been anaerobic. So hopefully you guys recognize the word aerobic, like aerobic exercise, meaning taking in of oxygen. These first protocells, those prokaryotes, would have been anaerobic. The reason we say that is they wouldn't have had any free oxygen to use. Oxygen was still bound in water and other compounds. It wasn't abundant in the atmosphere yet, so it would have had to not use free oxygen in any of its respiration or any of its life processes. It likely would have used infrared radiation from hydrothermal vents for energy. So the protocells would have been able to convert that IR radiation as its source of energy to fuel its chemical reactions and respiration inside of it. It would have taken, after those 3,500 million year old microfossils of prokaryotes, it would have taken millions of years for photosynthetic pathways to be developed. So these were not, remember we said these were heterotrophs, they're not autotrophs, they're heterotrophs. It would take millions of years for autotrophs to be developed. That's, millions of years sounds like a long time. You hear a million years and you think, wow, that's a really long time. But remember how many billions of years would have already passed. So essentially, millions of years in the grand scheme of billions is not a whole lot. But in these millions of years, photosynthetic pathways would have been developed, and those pathways would have released oxygen, free oxygen, O2, as a waste product. So it would have been the waste of early organisms. Oxygen would not have been essential for life, so it would be, junk, garbage, the byproduct of their respiration. These early photosynthesizers would have been what we think are cyanobacteria. Cyanobacteria, blue-green algae, cyan, right? The color blue-green. Cyanobacteria are photosynthetic, and they would have released oxygen as a waste product. This oxygen waste was released into the ocean and would have combined with the iron ions that were dissolved in the ocean water. And remember we said that the early ocean rusted? That's when that would have happened, when cyanobacteria developed the ability or evolved the ability to photosynthesize. Free oxygen would have combined with iron deposits or with iron ions and made deposits at the bottoms of the oceans. We can see evidence of these cyanobacteria, these early photosynthesizers, in rock formations. They look like small bubbles called stromatolites. Now you can see individual stromatolites here. Stromatolites are rock-like mounds that are formed by the secretions of cyanobacteria. There are still cyanobacteria, there's still blue-green algae on Earth today, and we can still see modern stromatolites being formed by cyanobacteria. This particular image that you're looking at is from Shark Bay in Australia, and each of these is a stromatolite formation created by cyanobacteria. That's how we knew what the fossil stromatolites were. We were able to compare them to modern stromatolites and realized that they were formed by the same process. Once the cyanobacteria released enough oxygen to rust the entire ocean, leaving those ore deposits on the ocean floor, oxygen was able to escape into the atmosphere. Now, this, to us, is no big deal. We're surrounded by oxygen and we're like, yeah, whatever, it's, not really much consequence. But oxygen is extremely reactive. This oxygen escaping into the atmosphere is a yay for modern organisms. Like, yes, it was absolutely essential that oxygen accumulates to modern levels for life as we know it to exist, but it's a big uh-oh for those early prokaryotic organisms that would have lived at hydrothermal vents because they were anaerobic. That means to them, oxygen is actually a poison. So it would have, killed them, but it allowed them to be replaced by modern organisms. Now, it didn't kill all of them. There are still places on Earth where oxygen is not abundant, and those very early archaebacteria still reign supreme as the dominant life form. This is where we go back to those hydrothermal vents. But as far as life near the surface of the ocean, that would have then killed off a lot of the anaerobic organisms that lived there. Once free oxygen was able to dominate in the atmosphere, it reacted with UV radiation. And this is really very important for us. So oxygen plus UV radiation. The UV radiation split oxygen molecules into free radicals of oxygen, and that allowed for a molecule that you guys call ozone. Or scientists recognize the major component of ozone is just oxygen. It happens to be 3 oxygen atoms bonded together, O3. So oxygen, O2, plus UV radiation made ozone, O3. That formed an ozone layer. The ozone layer is absolutely essential to protect Earth today and to protect life as we know it. UV radiation is very, very, very damaging to DNA. That's essentially how skin cancer happens. People's DNA is damaged by the UV radiation from the sun. Without the protection of the ozone layer, life on Earth couldn't exist as we know it because it would be essentially completely damaged by all of the UV radiation. So UV radiation would damage anything without ozone. So we can thank those early photosynthesizers for creating enough oxygen to allow the formation of an ozone layer. Probably, pretty good thing. That's why we're so concerned about there being holes in the modern ozone layer. After those early cyanobacteria, the archaebacteria, remember, all prokaryotic. We divided our kingdoms in the classification chapter into the bacterial kingdoms and then into the domain eukarya. Eukarya are all eukaryotic cells. If you don't remember what a eukaryotic cell is, I'll give you the brief reminder. Eukaryotic cells are more complicated, more complex. They are tenfold, so 10 times larger than prokaryotic cells. They contain organelles, a nucleus, mitochondria, chloroplasts. All of those things exist inside of eukaryotic cells. Eukaryotic cells form the basis for most of our multicellular organisms. Anything in the kingdoms that fall in domain eukarya, they rely on organelles, as well as infolded membrane layers and ribosomes, and they're more complicated, complex cells. There had to be a transition from prokaryotic dominating the world to then eukaryotic cells. There is a hypothesis made by a researcher out of Boston University. Her name is Lynn Margulis, and she established the theory of endosymbiosis, or the endosymbiont hypothesis. They're the same thing. They're just interchangeable words for the same hypothesis. And you remember what symbiosis is. Symbiosis refers to 2 separate species who live in relationship to each other. And this particular symbiosis that Lynn Margulis hypothesizes is a mutualism. It is a mutually beneficial relationship between free-living prokaryotic cells. Those 2 prokaryotic cells would have lived together in relationship where they both benefited. In fact, one would have lived inside of the other one. You're like, that's weird. Not particularly. You live right now in symbiosis with all kinds of things that reside inside of you in your intestinal lining, and we don't really think much of it. But what Lynn Margulis says is these two bacteria, these two different prokaryotes, eventually lived together for so long they were incapable of surviving apart from each other. So we'll take a look at a picture of what that looks like. So you'll notice here we have the many types of bacteria that young early Earth would have supported. There's a larger blue one, and then an oxygen-breathing bacterium, so an aerobic bacterium, and a photosynthetic bacterium. They would have lived inside of the larger, so here in this picture is our blue, larger bacteria. If they lived inside of them, inside of a larger cell, they would have been afforded protection from the environment by living inside something else. The thing that it's living inside of would have gained the benefit of the respiration of the oxygen-breathing bacterium, or for the photosynthetic bacterium, it would have gained the advantage of having the ability to harness light as a source of energy. If they lived together for so long, they could no longer live apart. Those two smaller bacteria that live inside of the larger one would eventually become organelles. The oxygen-breathing bacterium would have become mitochondria. You might remember from middle school, mitochondria is the organelle that, or mitochondria are the organelles that perform chemical respiration. So it's I think most middle school textbooks refer to it as the powerhouse of the cell. And knowing that that's, done by the respiration of oxygen, that's where that would have come from. The green photosynthetic bacterium would have given rise to the modern-day chloroplast, which I know you guys are familiar with as the site of photosynthesis. The evidence that we have that this actually could be a reasonable, plausible explanation. Mitochondria and chloroplasts have their own genetic material. The genetic material they have inside of them, it's DNA, but it's unrelated to the DNA within the nucleus of the organisms that they reside in. So it's separate DNA undergoing separate evolutionary influences. It's completely different than the host cell, or, the cell itself, since they're no longer hosts, since they live together. Mitochondria and chloroplasts also have a double layer of lipids. They have a plasma membrane of their own. Living cells, that first protocell, we suggested that it had a double lipid layer. That double lipid layer means that it was at one point free living. And mitochondria and chloroplasts reproduce at a rate unrelated to the reproduction of the cell. So it does not matter whether or not the larger cell is preparing for a mitotic division. So it doesn't matter if it's going to divide. Mitochondria and chloroplasts have their own schedule of division. Just like if they had been a free-living prokaryote, they would have divided, because that's essential for life, they would have divided at their own rate. So those are three pieces of evidence that give credence to Lynn Margulis's endosymbiont hypothesis. And this actually is a pretty good picture of that hypothesis, of how it could have possibly worked, that we ended up with our free-living, or our eukaryotic cells. Once eukaryotic cells were established, and the ozone layer was established, you see a massive explosion of biological evolution. You saw that massive explosion of evolution on your timelines that you guys created in class, where we said the atmosphere and hydrosphere were absolutely essential before any other events of early Earth could have occurred. And that's what we see here, just explained for us. That is essentially all of the material from this chapter. Remember, go ahead, take notes, ask questions, and your test is still on Tuesday.